Thanks so much, and thanks for inviting me to be with all of you. What I'm going to talk about in the next few minutes is sort of a fundamental problem I think that's facing everyone in data science right now, and that is a desire to deal with the FAIR principles, a recognition that the FAIR principles, which if you haven't heard of, we'll talk about in a moment, are just very difficult to deal with in the real world. And frankly, it's because the FAIR principles depend on metadata, which is obviously the key word for this whole meeting. And what I want to do is to give you an overview of really what we're doing at Stanford to try to address some of these problems. The whole business began, I would imagine, maybe 10 years ago, when people started to have concerns about the reproducibility of scientific results. And it sort of reached a head maybe 5 years ago when there was a classic study done by folks at a biotech company called Amgen, where they tried to reproduce some of the most classic studies in cancer biology and realized they couldn't get those studies to actually reproduce. They couldn't actually get the same results that were published in the literature. And people began to really worry, certainly in biomedicine, about whether the results of science actually meant anything at all. And that was one problem, particularly when it became clear that you couldn't actually go look at the data and understand the data to know what previous investigators may have done. Also, around the same time, became a real interest in secondary analysis of data. This is an award given at a conference called the Pacific Symposium on Biocomputing, where every year they give data parasite awards to people who've done the most exciting secondary data analyses. And of course, the challenge there is that you can't really understand what other people have done. You can't interpret their data unless you have metadata that describe what was done in the experiment, that explain what the data set looks like and how to interpret it. And fundamentally, people began to recognize that there was a need to come up with standards that would facilitate open interpretation of scientific results. Well, then came this landmark paper in Scientific Data, where a bunch of folks got together and said, you know, we need to come up with some principles for how to make data reusable and make it reproducible and to make it examinable. And the result was the FAIR guiding principles, basically an attempt to make data findable, accessible, interoperable, and reusable. And the FAIR principles really took hold because, for one thing, it was a great acronym. And secondly, it made a lot of sense. There was a lot of face validity to the idea that we should be able to have data that we can have online, that we can reinterpret, that we can understand. And suddenly everybody wanted to be fair. And in fact, it became absolutely crazy. People now are all trying to be as fair as possible. People claim to be fair when we don't even know how to measure fairness. And because of all of this emphasis on making data fair, we're in a situation where we really don't know how to assess whether the data that people are putting online actually mean anything. When you look at the FAIR guiding principles, they're pretty hard for most scientists to really understand. The data and the metadata are assigned global, unique, and persistent identifiers. Most scientists wouldn't know what that means. Data are described with rich metadata. Well, that's why we're all here today, I suppose. Metadata clearly and explicitly include the identifier of the data they describe. Metadata registered or indexed in a searchable resource, and so on. These are things which make a lot of sense, I think, to information scientists. The FAIR guiding principles turn out to be really hard for your typical bench investigator to understand. And so people have tried to make the FAIR principles meaningful primarily by building tools that can look at online data and determine whether they might be fair. Mark Wilkinson probably had the first system called the FAIR Evaluator, which would look at data sets and tell you whether they were fair. And there were lots of problems actually with whether this would actually work. Folks in the United States built something called FAIR Shake, kind of a great name for a program, but again, lots of problems in making it actually work. Currently, there's a big project in Europe developing Fuji. The Fair is Fair folks are trying to get that to become more popular. The real challenge is that as people try to be able to make data fair and understand how to build tools to assess this fair data, what we've discovered is that the systems that people are building to evaluate fairness really are not catching on. They've really struggled to find an audience because really the scientists who want their data to be fair don't know what to do with a report card that will tell them after they've already done all their work and submitted their data to some repository, whether they actually did it right, because at that point, it doesn't really matter to them. No one really wants to hear about problems with data sets that have already been uploaded. You want to be able to solve problems while you're actually in the midst of trying to deal with those problems. And the real problem is that there's no computable solution to determining whether a data set is fair in the first place. Well, why is that? Well, let's look at the fair guiding principles. We're not going to go through them in great detail, but look, most of the fair guiding principles deal with metadata, and that's okay. Some of the fair guiding principles actually deal with information about the repository where an investigator is going to be placing the data sets. And since most investigators don't really have any control at all over the repository, these fair principles really have no relevance to a scientist. But look at the ones that remain. A lot of them are dealing with things which are just simply not anything that a computer can deal with. Data sets are described with rich metadata. Well, what does it mean for metadata to be rich? Metadata use vocabularies that follow the FAIR principles. How do we know whether a vocabulary itself is following the FAIR principles? It sort of is a circular problem. Metadata are richly described with a plurality of accurate and relevant attributes. Well, what does it mean for anything to be richly described, and how do we get a computer to know whether something is rich or not rich? The metadata meet domain relevant community standards. Well, that's great, that's important, but how does the computer, again, know what are the relevant community standards and how whether they're actually applied in an appropriate way? So fundamentally, we're in a situation where we have no real ability to determine computationally whether the FAIR principles are being met. The FAIR principles depend largely on metadata. And just to take a small diversion, it's important to emphasize that when you look at the data that are in public repositories, the metadata typically are terrible. Most investigators view their work as publishing papers, not leaving a legacy of reusable data. Sponsors, management, all may require data sharing, but they don't usually encourage the use of funding that they're providing to actually pay for it. And being able to create metadata that are actually good in the first place is remarkably hard. And that's because almost all metadata usually are authored using spreadsheets. And here's an example of someone using a spreadsheet to author the metadata for a sample that's going into a repository in the United States called the Gene Expression Omnibus, which is a big database that looks at data related to how genes get turned on and turned off in the context of being able to understand different biological processes. And it's not surprising that people who use these kinds of spreadsheets screw up, because it's not really clear from the spreadsheet exactly what's required in order to create good metadata. It's not exactly clear from the spreadsheet what ontologies or controlled terminologies should be used to provide values for the information in the metadata. And as a consequence, when you look at the metadata that's online, here's metadata from a repository in the United States called Biosample. You'll see that you see attribute value pairs, which look reasonable. When you look closely, you see that the disease is specified as carcinoma hepatocellular. And in English, you wouldn't put the adjective after the noun. The ethnicity is given as Japanese, which is probably more a political identification than an ethnicity. The age is given as 57 year, not plural, and probably you really don't want a mixture of numbers and units in the same entry. And as a consequence, if you wanted to search for a middle-aged Asian person with hepatocellular carcinoma, the data that are in this record probably wouldn't show up, because the metadata simply don't have the specificity and the standardization to allow you to do that. In fact, we've looked at Biosample, looking at all the metadata records and looked at them computationally. And what have we determined? Well, 73% of the metadata values that are supposed to be Boolean actually don't have the value true or false. They have values like non-smoker or former smoker, but obviously not something that's an actual Boolean value. 26% of the integer metadata values don't get parsed into integers. They don't get parsed into numbers. They get values like JM52, UV, PGT. I don't know what these things mean. Pig. 68% of the metadata entries that are supposed to represent terms from biomedical ontologies don't actually represent terms from biomedical ontologies. They have values like presumed normal and wild type and things which we can kind of understand in natural language, but are not ontology terms. So if you're searching data sets to find particular Boolean situations, if you're searching the data sets for numbers, you're searching the data sets for ontology terms, the metadata are actually not going to be good enough to allow you to do that. And then when we look at the gene expression omnibus, look at a value in the metadata like the age of the subject. We don't just see age, we see age and age and age and pop age and age after birth and age in years and lots of different ways that people might determine the age or specify age in a string. Metadata authors need to use controlled terms so that if you're doing a search of data sets, you can, with confidence, enter a term like age and pick up all the data sets that mention the age of the subject. Currently, we have to basically imagine all the variants, imagine all the typos, and our ability to get information out of these online repositories is really constrained by the fact that we just can't get access to the data sets that we need. It means that the data parasites, it means that people who care about the reproducibility of science, all of them are stymied because our metadata are so terrible. Now, in science, people have tried to address this problem, and probably one of the best examples of this really goes back about 20 years, when the people who are working in the area of DNA microarrays, and this is a technology used in biology that allows you to understand how all the genes or some subset of genes get turned on and turned off under different experimental conditions. These DNA microarray data are stored in databases and people want to understand them. And the investigators who are working in this area got together and said, look, we need metadata that we can query that will allow us to understand what experiment was done. We want to know what was the substrate of the experiment, what was the particular microarray chip, what was the platform that was used, what were the experimental conditions. And what they did was they created a reporting guideline called Miami. Miami stands for Minimal Information About a Microarray Experiment. And what Miami was not really anything computable, but was a really important first step, which was an enumeration of all the things about a microarray experiment that you should say in your journal publication and that you should represent in your metadata so that people will understand what you did. And what that means is that journal editors said, okay, we expect all the Miami criteria to be captured if we're going to publish something in this area. And the data repository folks, the people, for example, who created the gene expression omnibus said, we want the metadata to include these values. And so Miami was a really important first step, describing a reporting guideline which really caught on and allowed the metadata to be very explicit and to be standardized in terms of what they were going to represent. Well, it didn't stop with Miami, at least in biology. People created minimal information about T-cell assays, or something called MIATA, minimal information required in the annotation of biochemical models, or Miriam, minimal metagenome sequence analysis standard, or MiniMES, or one of my favorites, MISFISHY. Basically, there are now hundreds of these reporting guidelines that have all come out of biology, where scientists said, we want our metadata to be standardized and to talk about explicit conditions of the experiment so that we can search data sets for these conditions and we can be confident that we've recorded everything that we need to know about the experiment in our publications and in our metadata. And if you go to fairsharing.org, which is a resource that Susanna Sansoni maintains at the University of Oxford, you can actually search for these reporting guidelines and understand how they're used to be able to describe the metadata of experiments that are done in very classic areas of science. Well, if we want to have fair data, we basically need to have good metadata. And if we want to have good metadata, What we really need are ontologies that can give us controlled terms. So rather than having a million ways of saying age, we want exactly one way of saying age so we can find those records and search for them. We want reporting guidelines like Miami that provide a list of all the standardized elements of an experiment that you want to comment on in order to know what was done when the experiment was performed. And most important, from my perspective, we not only want the ontologies and the reporting guidelines, we want technology that makes it easy to author these kinds of metadata in the first place. So we've been working at Stanford on an approach that we call CEDAR. CEDAR is the Center for Expanded Data Annotation and Retrieval, and we've been funded by the National Institutes of Health in the United States in order to make it easy for people to create good metadata. That means they're going to create metadata that use reporting guidelines that are endorsed by various scientific communities. And those reporting guidelines appear computationally as templates that allow scientists to fill in the blanks, basically, to author their metadata in a very convenient way using a web form. And when they fill in those blanks, they're not going to be putting in arbitrary terms. They're going to be putting in values whenever possible from scientific ontologies that give us standardized values for the fields that are in those templates. So when we think about the FAIR principles, we know that most of the FAIR principles that we're having trouble with are the ones that talk about community standards, that talk about the richness of the metadata, that talk about how domain relevant those standards are. And fundamentally, what we know is that the way for us to ensure that our metadata, as we enter them through the templates that are available in CEDAR, are addressing these FAIR principles, is to say what we really need at the core is something that we call a metadata template. Here is a metadata template that refers to the kinds of data that are stored in a repository called biosample. And in particular, these are specimens that are human that are being put into biosample. And this metadata template is a list of attribute value pairs, just in the reporting guideline I showed you earlier. You see sample name 56, organism Homo sapiens, tissue skin of body, sex male, and so on. And the important thing is that these metadata templates that are the centerpiece of the work we've done in CEDAR, reflecting the community standards that are referred to in the FAIR principles, basically represent the way that a group of investigators in some scientific community have gotten together, just like the Miami people, and said, we want metadata that are going to be describing the experiments that we do to adhere to the standards where we describe what are the minimal things you need to say about the experiment for someone to make sense of it. So basically, a template such as the one you see here can make metadata rich because it will include all of the information about an experiment that community of practice says is important. And then metadata will follow or will use standardized vocabularies that follow the FAIR principles, because when we create such a metadata template, we specify at that time what are the ontologies and vocabularies that we're going to use to provide values for each one of these fields in the template. And we know that by definition, such a template is meeting domain relevant community standards because it was created by a scientific community. And the scientific community got together and said, this is what we want our metadata to store. And so fundamentally, we can take a metadata template, a set of attribute value pairs that are standardized for a particular class of experiments, and we can use this as the basis for creating FAIR data, because we know that our metadata are going to follow those community standards. So basically, if we care about fairness, rather than trying to determine after the fact whether data sets are fair, what we really should be doing is making it possible to make sure that the data are fair from the very beginning. What that means is that in CEDAR, we have a system. I'm sure why my mouse keeps on clicking. CEDAR allows us to start out by creating a metadata template that is available in a computational form that allows us ultimately to fill in that template with experiment-specific information. And then when we have the template filled in to describe actual metadata, we can match that metadata with the data set that the template describes and upload the information to some appropriate repository. And by the way, One of the things that CEDAR does is it will keep a local database of all of the templates and all of the ways that templates have been filled in. And it uses that database when scientists are creating new metadata. And it can take advantage of previous metadata and learn patterns in those metadata to make it easier for the scientists to do their work. So let me explain what that means. When you log into CEDAR, what you see are a set of templates, also template components that you can use to build new templates. And you can see what you have before you are templates that you've created or templates that other people have shared with you, particularly scientific communities where they will settle on a particular template as being important for a particular line of investigation. And for example, you care about biosample human, that template I showed you a minute ago, we click on it, we say we want to populate it, and here we see the initial template for biosample human. And it has the attributes that we talked about earlier: sample name, organism, tissue, sex, and so on. If we want to fill in the template, then we click on any of the fields in our web browser, and we get drop-down lists that contain controlled terms. At Stanford, we have a repository of scientific ontologies that we call BioPortal, and BioPortal and CEDAR talk to one another so that when we click on a field in a template in CEDAR, you automatically get whatever ontology or whatever branch of an ontology has been specified by the developers of the template originally, to make sure that we're choosing the right set of correct terms for the field that we want to fill in. So when we select tissue, what will happen is that we'll get a drop-down list of terms that come from an ontology called Uberon, which is a standard ontology for vertebrate anatomy. And we make a selection from the drop-down list. And you'll notice that what CEDAR does to make our lives simple, to make it easy to fill in this form, is to sort the elements in the drop-down list. So the ones that appear most frequently in the previous metadata are at the top of the list. And we know that when the organism is human, by far and away, most of the tissue that is examined turns out to be blood. That's just something we can learn from the previous metadata. And to a lesser degree, we see tissues like liver and bone marrow and breast and lymph node and so on. So we make it really easy for users by anticipating what their selections are going to be. We try to use predictive data entry in the interface whenever we can. And frankly, we know that our competition is spreadsheets. And everybody likes spreadsheets because they're so easy to use. But as we saw earlier, the problem with spreadsheets is that their lack of structure typically leads to metadata that really are very difficult to search and very difficult to understand. So we fill in the fields using these drop-downs. Now, what we can do is we can tailor these drop-down lists in very explicit ways by looking at the context in which we're filling in the field. So for example, if someone has already specified that the tissue is lung, then we can automatically determine from our previously entered metadata, looking at the patterns in those metadata, that lung cancer is going to be the most commonly used disease associated with tissue that comes from lung, according to something called the disease ontology. And we know that chronic obstructive pulmonary disease is going to be a frequently entered disease. Squamous cell carcinoma of the lung is going to be important, and so on. And we know that if we change the tissue to say brain, by looking at the patterns in the metadata, we know that when the tissue is brain, we're going to be seeing other diseases that's at the top of our drop-down list: Parkinson's disease, CNS lymphoma, autistic disorder, melanoma that is metastatic to the brain, and so on. And so fundamentally, we try to make it really, really easy for scientists to fill in these forms, because it's really important for them to be able to use these forms to have standardized, complete metadata, or else we're not going to be able to adhere to the FAIR principles. So fundamentally, if we do want fair data, what we really want are metadata that use ontologies that give us controlled terms, reporting guidelines like Miami that provide a uniform structure and a set of attribute value pairs that we know are standardized and reproducible. And we need technology like CEDAR that makes it really easy to be able to create these kinds of templates or fill in these templates in the first place. So currently, almost all metadata are created using these spreadsheets. We love spreadsheets because they're easy, but we hate spreadsheets because they don't provide the structure that allows us to ensure that our metadata are going to become fair. And so we want to move away from the spreadsheet model and use these structured web forms. We don't want to allow users to specify any old term. We want them to use terms from ontologies to make sure that our metadata and therefore our data sets are searchable. So we want to be able to make sure that our terms are all standardized. And we don't want to try to give scientists report cards after they have already done their work and already done the uploading of their data sets to repositories and have them in a repository where they really can't change anything. We want to be able to solve the problem of creating good metadata and therefore ensuring fair data sets from the very beginning. And really, the only way that we can do that is to have tools like CEDAR that allow the data and the metadata to be fair at the time they're initially authored. So we look at those FAIR principles, we realize that the ones that are the hardest to deal with are the ones that are the vague principles that deal with richness and domain relevancy and so on. And we know what we really want to be able to do to come up with domain relevant community standards that are richly described, that use the right control terminology, is that we really need methods to create these kinds of metadata templates in the first place. And we've been partnering for the past couple of years with the GoFair initiatives. The GoFair initiative, as you know, emerged from the European Open Science Cloud, is an international organization, but has its greatest emphasis, I think, in Europe. We've worked with the GoFair folks to create what the GoFair people call metadata for machines workshops. So in an M for M or metadata for machines workshop, basically we bring together scientists from a particular discipline. We have them work very closely for 2 to 3 days. And basically their goal is to reach consensus about what are the essential metadata fields that need to be incorporated for their area of science and for the different classes of experiments that they do. So ultimately the goal is to come up with a reporting guideline like Miami for a different area of science and to render that reporting guideline, not as text, but as a CEDAR metadata template. So we have something that is not just text, but something that is computable. So fundamentally, we work very closely with GoFair to work with these scientists to generate these metadata templates that form the basis for our work in CEDAR, and that allows CEDAR to make data sets fair. And we've had a lot of interesting experience with these M4M workshops. Colleagues in Denmark who are working in the area of wind energy have been hosting metadata for machines workshops, trying to elicit the kinds of metadata that need to annotate the data sets that are being used to describe information related to wind energy experiments. And that's been extraordinarily successful. And in fact, the Danes are now thinking about trying to expand this to almost all areas of science that are funded by their government. We've been working very closely with an initiative called Vodan that is trying to acquire data about the spread of COVID in Africa. And we're currently creating metadata for machines workshops to elicit the metadata standards that are going to be used in CEDAR and installing CEDAR basically all over Africa with a goal of trying to be able to understand how to capture the metadata that are necessary in all of these observational studies and fundamentally to make those data sets fair. What I think is really interesting, the Netherlands Organization for Health Research and Development, known in Dutch as ZONMV, is very excited about this whole approach. And they've hosted metadata for machines workshops to develop standards that allow them to specify how they really want scientists whom they fund in their sponsored research to be able to manage data and make it fair. So fundamentally what happens is that ZONMV is hosting metadata for machines workshops, which are basically run by the GoFair folks. And these metadata for machines workshops, which are currently being done in the area of COVID, are allowing investigators to come together to define what they believe are the essential things to be saying about their data sets and to represent those essential elements of the metadata for their data sets using CEDAR. And then ZONMV is saying, well, look, if you want our funding you have to agree to use these metadata templates when you annotate your data and put your data in public repositories. So fundamentally, you're saying the metadata for machines workshops are the basis for determining how data will be made fair, and saying we want data to be fair, so you must use these templates. And now ZoneMV is not only doing this for their COVID work, they're looking at areas such as antimicrobial resistance and hosting metadata for machines workshops that are going to be looking at these other areas of science, trying to define new standards for metadata that CEDAR can then help scientists to use as the basis for describing data sets in a way that makes those data sets ultimately fair. So ultimately, my belief is that fairness is really important. Fairness is essential if we care about the reproducibility of science. Fairness is critical if we want to be able to reanalyze existing data sets and make secondary analyses that will lead to new discoveries. If we want to merge our data with someone else's data and see whether the merger of those data sets can give us more power to make new observations. And fundamentally, we can't have fair data that allows us to do all those things until we have good metadata. And those good metadata will require a metadata structure that is standardized using a template based on reporting guidelines. It's going to require controlled terms that come from standardized ontologies. And it's going to basically require technology like CEDAR that really makes it easy for investigators to annotate their data sets in ways which assures that the data sets are standardized and searchable and that the data are fair. And so right now we're in a situation where we don't want to be spending a lot of energy trying to build yet more fair evaluators to determine whether data are fair after the fact. We really want to be putting more energy into being able to make it easy for scientists to create data sets that are fair upfront. And I think one of the exciting things about your workshop today is that you'll be exploring metadata in a variety of venues. And it's really important to remember that metadata are not really just arcane descriptions of data sets. They're not just the domain of information scientists. Metadata are absolutely critical if we want to do science right. And if we care about reproducibility, if we care about new discoveries from secondary analyses, then we really need to be able to think about templates, ontologies, and technologies like CEDAR that can put it all together so that scientists can make their data fair from the very beginning. And let me stop there and let me thank you for your attention.